Hello again, everyone. My name is Nate. If you've clicked on this video, that means you clicked on the spliced out version of the electron transport chain and oxidative phosphorylation. I will, of course, include this video in my longer full-length metabolism video, but if you just wanted to watch the content information for the ETC and ATP synthase, this is where you'll find it, in case you didn't want to click through the full video. I'll just mention, of course, again, that I do have my website called MCAT. You can find the link for it in the description. You can find my other free resources, my tips, my videos. I also do offer tutoring through that website, yada, 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 so on and so forth. All of that can be found on my website. It's linked in the description. That is also where you can find the notes that I've taken to support the info in this video. Basically, I've tried to actually write out all of the notes that you need to know. That way, whenever you're watching the video, all you have to do is just sit back and watch. You don't have to actually take notes, and you can prioritize understanding what I'm saying rather than writing it down. So the electron transport chain follows the Krebs cycle. At this point, we have produced a whole bunch of electron carriers that we now need to do something with. Many of the earlier steps will have produced ATP directly. We can just immediately use that ATP. We don't have to send it through anything else. And we have now completely used up that carbon chain, so we're not going to be using any molecule like acetyl-CoA or pyruvate for anything anymore. Now at this point, we are just going to be taking these electron carriers and we're going to be sending them through the electron transport chain. And what this is going to help us do is create what's called a proton gradient. We're going to be taking a lot of protons, hydrogen atoms from the inside of the mitochondrial matrix. We're going to be pumping them out of the mitochondrial matrix, creating a gradient that we'll be able to take advantage of later. So just to reiterate, what is the point of the ETC? We have directly built ATP in both glycolysis and the Krebs cycle. Like I mentioned earlier, our cells can just immediately use that. We don't have to do anything else with it. For example, if we're just doing anaerobic respiration, those two ATP that we build in glycolysis, we can immediately use. But especially in aerobic respiration, we've produced a ton of these electron carriers. That these electron carriers can't directly be used as energy. We can't use NADH to catalyze the production of a protein, for example. We take advantage of their electrons to produce more ATP. And this ATP is therefore what we're going to be using to actually be doing things like building molecules, proteins, so on and so forth. This is exactly what the electron transport chain is for. Looks like a pretty intimidating diagram, and it is one of the more complex parts of metabolism, but that's the whole point of this video. We'll dive in, explain everything that you need to know, make sure everything makes sense. Kind of like we did before the pyruvate dehydrogenase complex, we need to kind of talk about the structure of the mitochondria in order for the full depth of the ETC to actually make sense. We're going to start with the matrix. You'll notice that I'm boxing the terms in the picture as they start to show up. The matrix is the very inside compartment of the mitochondria. This is where most of our metabolic processes that occur in the mitochondria are going to be housed. So the PDC, the Krebs cycle, all of those enzymes are going to be inside of the mitochondrial matrix. The mitochondria actually has two separate membranes. The first membrane is the inner membrane that we're going to start off with. This is the one that surrounds the mitochondrial matrix. And the electron transport chain and ATP synthase, both of these are enzymatic complexes that we're going to be talking about today. They are both located directly on the inner mitochondrial membrane. This membrane is completely impermeable, including to these protons that we're making this gradient with, meaning that nothing really can pass through without us either opening a channel or having some sort of enzyme to help us move it across. One specific structural component of the mitochondria I just wanted to mention really quickly, the cristae are the individual little folds that we see on the inner membrane. They're just there in order to increase the surface area of the inner mitochondrial membrane. The intermembrane space is what we hit whenever we leave the inner membrane, working our way out of the mitochondria. This is the main area where that proton concentration that we're creating is going to develop. The intermembrane space sits in between the inner and outer membranes, which is why it's called the intermembrane space. The outer membrane is the one that's going to be separating the mitochondria from the rest of the cell, the cytosol that's around it. It is slightly permeable, including to some protons. So we are going to see a little bit of that proton gradient seep out into the cytosol. But the outer membrane in general, for what you need to know right now, it's basically the cap around the mitochondria. Another quick little interesting note, mitochondrial DNA is circular. It has its own DNA that contains a bunch of genes important for metabolism and apoptosis. Just as a reminder, the eukaryotic genome inside of the nucleus is linear, not circular. The mitochondria actually used to be a bacteria that a now eukaryotic cell once engulfed. They realized how cool of a setup it was in a symbiotic relationship. This whole process is called endosymbiotic theory. Led to a whole bunch of genetic and metabolic diversity. Long story short, your mitochondria actually has its own DNA that replicates itself and has its own genes. It is circular, and it only comes from your mother. This is called maternal inheritance. So if your mother has some sort of metabolic or apoptotic gene disorder in the mitochondrial DNA, all of her children will also have that disorder. Not super important for the exact processes going on in the mitochondria that we're going to be talking about today, but very important for the purposes of the MCAT. Wanted to mention it right now. Now to actually dive into the electron transport chain, just to start introducing it, like I mentioned earlier, it is located directly on the inner mitochondrial membrane. And since we are inside of the mitochondria, this is considered an aerobic process, especially because of a step that's going to happen at complex 4 at the end. The main goal of the electron transport chain, like we've been alluding to earlier, is to receive these electron carriers from all of these metabolic processes, both from glycolysis in the cytosol and the PDC and Krebs cycle in the matrix. And we're going to be using these high energy electrons that they carry to pump protons out of the matrix and into the intermembrane space. This is going to create a concentration gradient where the protons really want to go back into the matrix, but because that inner membrane is impermeable to these protons, they're going to have to pass through a channel in order to do so. We'll talk about why that's important whenever we get to oxidative phosphorylation. We're going to take advantage of that fact. The ETC is mainly going to be on when we are in aerobic conditions, when we need ATP. Basically, the ETC is always on whenever we need to be producing ATP. Not too many specifics you need to know beyond that. This is pretty much on all the time, but we're just using this as a good time to finish up our discussion of aerobic respiration. The enzymes and complexes that you need to know, like every other process, there's going to be more on these in the next few slides. But to start, complex 1 is going to be the place where NADH dumps off its electrons. NADH is only going to give its electrons to complex 1 and then it's done. Complex 2 is going to be the exact same, but specifically for FADH2. Complex 3 is going to be receiving the electrons from complexes 1 and 2. They're going to be passed to a molecule that we'll talk about later in order to actually get from complexes 1 and 2 to complex 3. And then complex 4 is going to be receiving those electrons from complex 3. It's going to pump some protons, and then it's going to pass its electrons onto oxygen in order to finish the cycle. This is why you might have heard oxygen be described as the final electron acceptor, because it is basically allowing complex 4 to reset itself, pass on the electrons that it doesn't need anymore, so that it can accept new ones in a new cycle. Of course, it's going to be promoted by high amounts of electron carriers and low amounts of ATP, and it's going to be inhibited by low amounts of electron carriers and high amounts of ATP. Basically, if we have a whole bunch of ATP, we don't need to run it anymore. If we don't have a lot of ATP, we'll kick this in to high gear. So before we actually start talking about how the electron transport chain works and all of these enzymatic complexes work, there's two really important molecules that you need to basically be able to recognize and understand the basics of. The first one is called ubiquinone. This is also known as coenzyme Q, CoQ. This is a molecule that is only going to be taking the electrons from complex 1 and 2, and it's going to be giving these electrons on to complex 3. That's all it does. It simply passes electrons from either of those first two complexes onto complex 3. This is the basic structure of ubiquinone. You don't need to be able to draw this from scratch, but I do want to point out this little structure right here. This is called a quinone, which is why that term quinone is in the name ubiquinone. You should be able to recognize a quinone, and we're going to be using this to talk about the redox reaction that ubiquinone is going through. So when coenzyme Q has received the electrons from either of those first two complexes, it is reduced. And at this point, we call it ubiquinol instead of ubiquinone. You sometimes see that now abbreviated as CoQH2. So notice in this ubiquinone structure, the structure up here with ketones in this ring, the only thing that we've done in this reduction is that we've turned those ketones into alcohols, which is why we end up with the naming ubiquinol instead of ubiquinone. So then whenever coenzyme Q then passes its electrons on to complex 3, or it's just waiting to receive electrons, basically it doesn't have the electrons, it is oxidized, and now we just call it ubiquinone or coenzyme Q. Kind of like I alluded to a second ago, in organic chemistry, the naming "-one" means there's a ketone involved, which you can see that in ubiquinone. The ending "-ol" means there's an alcohol. You can see that in ubiquinol. Like I mentioned, you don't need to memorize the structure. You just need to memorize a quinone and know the difference between ubiquinone and ubiquinol. The next molecule that we need to talk about is called cytochrome C. It's sometimes abbreviated as Cyt C. This is going to have a similar job to coenzyme Q, where it's simply going to be responsible for giving electrons from complex 3 to complex 4. That's all it does. It's passing electrons from one place to another. A few important notes about cytochrome C that the MCAT kind of likes to hone in on. Cytochrome C does contain heme, which is an iron-containing molecule that specifically is a coordination complex between iron, which in this case will be serving as the complex ion if you know anything about coordination complexes, and porphyrin, which is going to be the ligand. It's that ring structure that surrounds that iron. So cytochrome C contains this heme structure. Again, you don't need to know the exact structure of heme or cytochrome C. You largely just need to be able to identify this porphyrin ring and know that heme basically is iron with this surrounding porphyrin ring, and that cytochrome C contains heme. Sounds sort of complex, but like I said, you just need to be able to recognize it. You don't need to memorize the structures. Cytochrome C, additionally, is actually going to be on the intermembrane side of the inner membrane. So basically, the inner membrane is going to have one side that faces the matrix, another side that faces the intermembrane space. Cytochrome C just happens to reside on the intermembrane side. Not super important reasons as to why for the purposes of the MCAT, but that's just another thing that I recommend at least just having a knowledge of. So now that I've introduced the ETC as well as these important molecules that are going to help us along the way, we get to actually talk about how the electron transport chain works. I'm going to give a basic overview of pretty much how every single complex will operate. They have the same 3 steps. And then we're going to specifically talk about every single enzymatic complex as well. So there's going to be 4 different complexes that are each going to operate with the exact same 3 steps, with one exception. We'll talk about that in a second. First, the complex will receive electrons, 2 electrons specifically, from NADH or FADH2. These electron carriers can come from any metabolic process. Now that the complex is reduced, the complex now has the ability to pump protons out of the matrix and into the intermembrane space. The complex has now completed its job. Its job is to pump protons, and now that it's done, it needs to reset itself. It needs to lose these electrons before it can repeat the steps in 1 and 2. The complex is then going to give up these electrons to some sort of other electron carrier, and therefore is going to be ready to receive electrons from any other source, specifically the electron carriers themselves in their reduced forms. So just to help you out with the color-coordinated system that I'm building right now, you'll notice that when something is oxidized, I have it in red. When something's reduced, I have it in this bright blue. You're only really going to see this on this slide down here when I talk about the naming. We'll get to that later. And then additionally, notice that the arrows are also colored as well. Sorry if you're colorblind. I was trying to make this as colorblind friendly as I could, but I didn't really have any options beyond orange, yellow, and green at this point. So the orange arrows over here will demonstrate that step 1. The yellow arrows will demonstrate step 2, and then the green arrows will demonstrate step 3 for every single complex. This will be reiterated on the next slides when we go through every single complex. So these electrons that we are receiving from NADH and FADH2, they are so high in energy that they can actually keep being passed between several different complexes before they have been exhausted. This is why the electrons from NADH can go to 3 separate complexes, as well as FADH2. They are going to start in different places, but they are all going to get past complexes 3 and 4. That's how high energy these electrons are. Additionally, to start helping with the naming convention of the complexes, they're going to have basically three different parts. There's going to be the oxidized electron carrier from step 1, followed by a hyphen with the reduced electron carrier from step 3. Basically, the thing that gave the electrons to the complex and then the thing that's taking it right after. And then finally, oxidoreductase. Notice that the oxidized electron carrier from step 1 is first, which is why in oxidoreductase, oxido is first as well. The oxidation is first, the reduction is second. Just as an example, the easiest example to come up with is complex 1. Complex 1 is also called NADH coenzyme Q hydride oxidoreductase. So notice that NADH is going to be giving its electrons to complex 1 and therefore is oxidized. NADH is oxidized into NAD plus at complex 1. And therefore, that's why it shows up first. Coenzyme Q is going to be the reduced electron carrier that we produce at complex 1, which is why it's second, and that's why you also say that it is the reductase portion. Just to show you, NADH is oxidized, which is why it follows that first prefix. Coenzyme Q with that hydride is the reduced form, that's why it follows the reductase. Some of these complexes are also going to have a simplified name, which maybe they chop off half of this naming convention. We're going to list and talk about all of them on the next slide. So now to pick up with every single complex of the electron transport chain. Again, sorry, everything's color coordinated still with that orange, yellow, and green system with the 3 steps. So my apologies if you have protanopia or deuteranopia. I'm pretty sure those are the two that would struggle with different colors of yellow and orange and red. Could be wrong. I'm working with like 4-year-old memory from my psych and social class at that point. So my apologies on that. But to begin with complex 1 is going to be here in purple. This is called NADH coenzyme Q oxidoreductase. That's basically telling you that NADH is oxidized, coenzyme Q is reduced, like we talked about on the last slide. It is also called NADH dehydrogenase. Remember that a dehydrogenase in metabolism is an enzyme that is helping us with a redox reaction. So NADH dehydrogenase basically means that we are just oxidizing NADH. We're going to be reducing the complex itself in doing so. So to talk about every single step. Step 1 in orange, NADH is going to be giving its 2 electrons to complex 1. NADH will be oxidized into NAD+. In step 2, the yellow arrow, now that the complex is reduced, complex 1 is going to be able to pump 4 protons. Every single time complex 1 receives electrons from NADH, it's going to be able to pump 4 protons at a time. This will be important later. Then finally, in step 3, the green arrow, in order to reset itself to receive more electrons from NADH, it's going to pass on the electrons to coenzyme Q. So now that complex 1 has passed its electrons onto coenzyme Q, it is back in its oxidized, waiting for electrons form, and it is ready to receive more electrons from NADH. Complex 2 is called succinate coenzyme Q oxidoreductase. And this naming is kind of weird, so we're going to talk about it here in a second. Basically, remember that from the Krebs cycle that we talked about in the last video or the last portion, depending on which form of my video you're watching, that the oxidation of succinate forms fumarate, and in doing so is going to produce FADH2. So just as a sort of shortcut, the succinate dehydrogenase enzyme is actually going to be the exact same thing as complex 2. Basically, succinate is going to form fumarate and FADH2 right at this complex so that FADH2 can be right there in the exact same spot in order to actually give the electrons to complex 2. Sort of complex. Don't worry about it too much. I'm just trying to explain why you might see some weird naming of complex 2, and this is just another thing that the MCAT might test on. Succinate is being oxidized into fumarate. That is giving complex 2 and succinate dehydrogenase the electrons that it needs to actually get this chain reaction going. I'll reiterate and re-explain complex 2 here at the end whenever we're done with it, just so you fully understand what's going on. Just to finish off the naming convention, succinate dehydrogenase, it's the same thing as complex 2. It can also be called succinate coenzyme Q oxidoreductase. We are oxidizing succinate, which is in the middle going to help us produce FADH2, which is going to then pass on its electrons back to the complex and then on to coenzyme Q. Very confusing. Like I said, let's just go through the steps, and by then it should make sense. So before we pick up with step 1, succinate has been oxidized into fumarate. So now that there's been an oxidation, that's going to allow a reduction to occur. We are going to reduce FAD into FADH2. as you can see in this arrow right here. And now that we have produced FADH2, it can immediately turn around and give those electrons back to complex 2. So now that it has given its electrons to complex 2, FADH has given its electrons to complex 2 through this weird succinate path, complex 2 is actually not going to pump any protons. That's why there's no yellow arrow on complex 2. So it's simply going to pass its electrons onto coenzyme Q now in step 3. Basically, complex 2 is going to be skipping over step 2. The reason for this is the electrons in FADH2, you can basically think of them as less efficient, less high energy than the electrons in NADH. Complex 2 is used as the entry point for those electrons, but those electrons don't have enough energy to fully pump 4 protons at complex 2 like we did with complex 1. FADH2 electrons are weaker, which is why complex 2 is not going to be pumping protons. Just to reiterate what is going on with complex 2. Complex 2, which is essentially the same thing as succinate dehydrogenase, it's going to take succinate, it is going to oxidize succinate into fumarate, which is going to help us produce FADH2 in that redox reaction. Now that we have that FADH2, we can immediately turn that around and give those electrons back to complex 2. Now that complex 2 has the electrons, it doesn't necessarily want to use that energy yet, so it's going to pass on those electrons to coenzyme Q, forming the reduced form. You could also call it ubiquinol, and you should be able to use those interchangeably. Now that we have formed the reduced ubiquinol, ubiquinol being the reduced form of ubiquinone, we can then pass the electrons onto complex 3. Complex 3 is going to be receiving the electrons from this ubiquinol that we've just produced. It's going to be pumping some protons, and then it's going to be giving the electrons off to cytochrome C. So it's receiving electrons from ubiquinol, it's giving electrons at the end to cytochrome C in step 3. It's because of this that sometimes we shorten it. We just remove the coenzyme Q and oxidation part of the naming. We just call it cytochrome C reductase because it's the enzyme that will essentially be reducing cytochrome C so that it can pass on electrons to complex 4. So in step 1, coenzyme Q, the reduced form, ubiquinol, is going to be giving its electrons to complex 3. Notice it's still 2 electrons. This is the orange arrow that we're talking about here. This is going to allow complex 3 to pump 4 protons. And then complex 3 will then pass on the electrons to cytochrome C. This allows complex 3 to reset itself so that it can accept more electrons from coenzyme Q. Now that cytochrome C has the electrons, it is going to be passing those electrons onto complex 4. Because of this, all that complex 4 really is doing is it is oxidizing cytochrome C, which is why it's simply called cytochrome C oxidase. So in step 1, the orange arrow, cytochrome C is going to be reducing complex 4. Cytochrome C itself is oxidized. It's lost those electrons now. As a reduced complex, complex 4 will then pump only 2 protons. It's basically exhausting any of that remaining energy left over from these electrons. It can only pump 2 at a time. And then finally, in order to reset itself, complex 4 is going to be passing off those electrons onto oxygen, basically allowing us to reform water. This is why oxygen is often called the final electron acceptor. It's allowing complex 4 to get rid of those exhausted electrons so that it can accept more from cytochrome C and pump more electrons. So now just to talk about the path of electrons that both go through NADH and FADH2. So the electrons from NADH, notice I have entirely covered up complex 2. The electrons from NADH are going to start off at complex 1. The electrons are then going to go from complex 1 to coenzyme Q. Coenzyme Q is going to pass them on to complex 3, and then through cytochrome C, complex 4 will then receive the electrons. The electrons from NADH are going from complex 1 to 3 to 4. Notice we have entirely skipped over complex 2. And you'll also notice that that means that the electrons from NADH will have allowed us to produce 10 protons. If you're counting up how many electrons have been pumped at each of these steps, we have 2 at complex 4, 4 at complex 3, and 4 at complex 1. So those electrons from NADH are going to help us pump 10 protons. Every single NADH will pump 10 total protons. When we get to oxidative phosphorylation, we'll talk about why that's important. If you remember what we talked about earlier, remember that FADH did not actually start at complex 1. FADH came through that succinate dehydrogenase system at complex 2. So now we're going to fully cover up complex 1 because FADH and its electrons never touch complex 1. They're going to start at complex 2. They're going to go through coenzyme Q to complex 3, which will then go through cytochrome C on to complex 4. So the electrons from FADH go from complex 2 on to complex 3 and then to complex 4. Notice that because we skipped out on complex 1, the electrons from FADH are only going to be able to pump 6 protons per FADH2. This is what I was talking about earlier, that the electrons, you can kind of think of them as weaker, lower in energy, whatever works for you. Just note that FADH2 can only pump 6 protons per molecule. NADH is going to be able to pump 10. The last thing to mention, just to kind of start connecting the dots from biochemistry to some of your other regions of content, like general chemistry, for example, remember that the pH of a solution is determined by the hydrogen concentration. And we are shipping hydrogen out of the mitochondrial matrix and into the intermembrane space. So because of that, we now have a growing hydrogen concentration in the intermembrane space, meaning that it is going to have a lower, more acidic pH than the mitochondrial matrix. So conversely, if we somehow completely turn off the electron transport chain, that means there's not going to be as many protons pumped as usual, as many hydrogens pumped as usual. The intermembrane space will be slightly more basic than usual, which also means that the actual mitochondrial matrix will be slightly more acidic than usual. That's something the MCAT does a lot. They'll ask you what happens if you inhibit something. One of the ways they commonly do it is by asking about the pH of either side of the intermembrane if the electron transport chain is inhibited. So just to start actually summarizing the electron transport chain before we dive into oxidative phosphorylation and some of the other important things to know about the mitochondria, we're just going to summarize the electron transport chain just by reminding you that it is actually part of the mitochondrial inner membrane. And because it's inside the mitochondria, and obviously that last step with complex 4, it is aerobic, meaning it requires oxygen. I'm going to make a comment about this again whenever we get to complex 4. To summarize each of the complexes, complex 1 is going to oxidize NADH. It is going to then pump 4 protons, and it is going to reduce a ubiquinone into ubiquinol. Complex 2 is going to do the exact same thing, but instead of oxidizing NADH, complex 2 will oxidize FADH2, and it is incapable of pumping protons. It's also called succinate dehydrogenase because of all of that chain reaction that we talked about earlier, where we'll first oxidize succinate to form FADH2, then we're going to oxidize FADH2 and give those electrons to complex 2, and then on to ubiquinone to form ubiquinol. Again, very importantly, complex 2 does not pump protons, and it is going to be the point of entry for FADH2 instead of NADH, but we are forming the same ubiquinol molecule as complex 1. Complex 3 will be oxidizing this ubiquinol that we got from complex 1 and 2. It is then going to pump 4 protons, and then it's going to pass those electrons on to cytochrome C. Cytochrome C is then going to be oxidized by complex 4, which will pump the last 2 protons that we can get out of those electrons, and then it will use that final electron acceptor in oxygen, thus also emphasizing why it's important that the electron transport chain is considered aerobic. It is going to reduce oxygen into water. So just to kind of mention this again, if we are in an anaerobic environment, meaning oxygen is not present, that means that complex 4 is going to get stuck with these electrons. It's not going to be able to get rid of them. So if it's stuck with the electrons, let's start working our way backwards a little bit. If complex 4 is stuck with electrons, that means cytochrome C is not going to have anything that it can pass its electrons to. So there's going to be a buildup of reduced cytochrome C, which means that cytochrome C is no longer going to be able to accept electrons, which means that complex 3 is no longer going to have anywhere to pass those electrons onto. And so complex 3 is going to build up in the reduced form, which means that complex 3 will no longer be able to accept electrons. And then we're going to have a buildup of ubiquinol. Ubiquinol is not going to have anywhere to give its electrons because complex 3 is already reduced. As you can see, basically, it's really important that we have oxygen because if we don't, then the electron transport chain is going to be completely inhibited. Additionally, just as a reminder, the electron carriers NADH and FADH2, they can come from any metabolic process, glycolysis, the pyruvate dehydrogenase, Krebs cycle, whatever. It doesn't really matter. The electron transport chain doesn't discriminate. It's just going to see electron carriers and it's going to use them. The electrons from NADH will go from complex 1, then on to 3 and 4. It is going to be able to pump 10 protons per NADH. Those electrons can power the pumping of 10 protons, I should say. The electrons from FADH2 will skip over complex 1 and start at complex 2, then passing electrons onto complex 3 and 4. This is going to allow the electrons from FADH2 to pump only 6 protons per FADH2. Just as a reminder that FADH2 is not as efficient of a proton pumper as NADH. This will be important later whenever we get to oxidative phosphorylation. This is a Gen Chem concept that I, quite frankly, don't want to talk about right now because I don't really like Gen Chem, but that's okay. I'll still mention a little bit about it. So reduction potential, that's basically this idea of how badly does something want to be reduced. In this case, how badly do the complexes want to receive electrons? So in order to keep passing the electrons down the chain, the next complex has to have a higher reduction potential than the one it's receiving electrons from. Basically, the reduction potential will increase as we get further into the electron transport chain, which is just what allows complex 4 to pull electrons from complex 3. And finally, just to quickly compare what's going on with the matrix and the intermembrane space and technically cytosol at this point, because as a reminder, the outer mitochondrial membrane is permeable to hydrogens. So some of the effects that we see occurring in the intermembrane space will also be slightly occurring in the cytosol as well. Since we are pumping protons out of the matrix, that means the matrix is going to have a lowering hydrogen concentration, which means that it will have a raised, more basic pH. The intermembrane space, since it is receiving those hydrogens, those protons from the matrix, it's going to have a higher hydrogen concentration, which means it will have a lower, more acidic pH. So remember earlier when we talked about how it takes energy to move large molecules across membranes? This was really important for the PDC because we had to do it with pyruvate. We've already talked about this with pyruvate earlier, but I had mentioned that you don't really need to know the exact way that pyruvate makes its way from the cytosol to the mitochondrial matrix. You don't really need to worry about that. You just need to know that it has to get moved and that we're going to lose potentially a little bit of ATP production in doing so. The NADH that is produced in glycolysis is stuck in the cytosol, and we are going to have to know how that makes its way inside of the matrix. And then just as a reminder, why is this important? Because in the electron transport chain, all of those sites where NADH and FADH2 bind to the electron transport chain are all inside of the mitochondrial matrix. So electron carriers being in the intermembrane space or cytosol are not at all helpful to us until they make their way inside of the matrix. So now we get to talk about how that actually happens. Basically, NADH is going to have two different systems in which we can get it inside of the mitochondrial matrix. There's two different shuttles, and this NADH, as a reminder, is coming from glycolysis. It could theoretically also come from any other process that's producing NADH in the cytosol. Basically, any NADH that's in the cytosol needs to make its way into the matrix. Here's the two ways that we're going to do it. The main thing that I want you to take away from this slide for the purpose of the MCAT is that these shuttles allow us to move the electrons from NADH from the cytosol into the matrix for the electron transport chain. But because we're having to move it across a membrane, that is potentially going to decrease our ATP production. The first shuttle that we're going to talk about is the one on the left, the glycerol 3-phosphate shuttle, G3P shuttle for short. So this enzyme that's sitting inside of the cytosol, kind of right around the mitochondria, called G3P dehydrogenase, since it's a dehydrogenase that, as a reminder from all of our other metabolic processes, that means it's going to be doing a redox reaction of some sorts. Here specifically, it is going to be oxidizing NADH. It's going to give those electrons to a molecule called DHAP, and it's going to reduce this DHAP into G3P. From here, G3P has a shuttle that its job is actually to carry G3P from the cytosolic side of the inner membrane into the matrix. So G3P can just get shuttled across the membrane by this protein shuttle that exists, the G3P shuttle. Then G3P dehydrogenase on the inside of the mitochondrial matrix will then do the reverse reaction of what we did in step 1. It's going to take G3P, it is going to oxidize it back into DHAP, allowing us to reduce FAD into FADH2. So notice that the electrons, you can kind of think of it as the electrons have lost a little bit of their potential energy. So instead of us giving the electrons back to NAD to form NADH2 inside the matrix, we had to give them to FAD instead. And remember, FAD is not as efficient of an ATP producer as NADH. We'll talk about that later. Essentially, all that's happening is we are passing the electrons from a cytosolic NADH to a matrices FADH2. This is going to cause us to lose a little bit of AT production since NADH pumps more protons than FADH2. This shuttle in general is very quick, but it is going to decrease our ATP use. So kind of like how we talked about with the PDC and fermentation, there's some scenarios where we need ATP fast. The G3P shuttle is one of those scenarios. We're going to prioritize using the G3P shuttle because it's faster at producing ATP, just slightly decreases our yield a little bit. This is what the actual reaction looks like. It's pretty complex. Like I said, I only recommend just understanding the main takeaway, understanding this idea that basically what we're doing is we are passing the electrons from a cytosolic NADH to a matrices FADH. And that's how we can essentially get the electrons from cytosolic NADH into the mitochondrial matrix for the electron transport chain. The malate aspartate shuttle, the one on the right, is going to essentially be using NADH to reduce oxaloacetate into malate. If you remember from the Krebs cycle, this is kind of the opposite of that last step in the Krebs cycle. From here, this reduced malate has a protein shuttle similar to G3P that basically can just help it move inside of the matrix. Conformational shift as soon as malate binds, that just kind of throws it on the inside. Once inside of the matrix, malate is then oxidized back into oxaloacetate. Redox pairing, if something's oxidized, that means we get to reduce something else. And notice here, we get to reduce NAD back into NADH. Unlike the G3P shuttle, the malate aspartate shuttle actually reforms NADH in the mitochondrial matrix. So this shuttle, the malate aspartate shuttle, or MA shuttle for short, because I'm lazy, is actually going to be more efficient in ATP use because we're not really losing any ATP yield, moving these electrons from the cytosol into the matrix. But it is, however, much slower. So we're only going to be doing the MA shuttle whenever we don't necessarily need ATP quickly and we can prioritize efficiency. Basically the exact same idea of how we run the PDC, when we can prioritize efficiency, we run fermentation when we need things fast. That's the same kind of compare and contrast that we have between these two shuttles. Again, the main takeaway that you need to know is that these shuttles are allowing us to move the electrons from cytosolic NADH into the matrix so that the electron transport chain can use them. The G3P shuttle is going to specifically give those to FAD, so we're going to lose a little bit of ATP yield. The MA shuttle can just reform NADH inside the matrix. One's faster, the other one's slower, but more efficient. So now at this point, we've produced this proton gradient. We've taken these electron carriers and we've produced this elusive proton gradient that we're going to be doing something with. So now the next question is this process right here, oxidative phosphorylation. How in the world are we getting from a proton gradient to actual ATP? Now that we have a proton gradient, how are we going to use it? The answer to that is this protein called ATP synthase. In a process called oxidative phosphorylation, you might have heard that term before, ATP synthase is essentially going to couple this favorable movement of protons down their concentration gradient back into the matrix to the production of ATP. We're going to talk about how that works on this next slide. So oxidative phosphorylation, it is also, just like the electron transport chain, going to be located on the inner membrane of the mitochondria. Basically, what's going to be happening is we have this enzyme called ATP synthase. ATP synthase basically has this big channel that's going to allow these protons specifically to move back into the matrix, which is a movement that the protons want to do. They want to go from high concentration to low. This is a favorable movement that with enough protons moving, with enough essentially potential energy generated by the movement of these protons, we can essentially force the reformation of ATP. we can force ADP to accept another phosphate, forming ATP. And of course, we're going to be able to use that ATP for whatever we want. We are allowing protons to move back into the matrix, and we're coupling that movement to the formation of ATP, essentially. The first subunit that you need to know just a little bit about is the F0 or F0 subunit, whatever you want to call it, that's fine. This is a multi-subunit transmembrane protein. You don't really need to know too much about that, but basically it's this transmembrane protein channel that just opens and only is going to allow protons through. So the inner membrane is impermeable to nearly everything, and this channel is going to open just for protons to slide through. Now that protons are sliding through, the F1 subunit, the actual catalytic subunit that is inside of the matrix, it's on the matrices side of the inner membrane of ATP synthase. The movement of these protons through the F0 subunit is basically going to cause this rotating conformational change in the F1 subunit. And this rotation essentially is going to slap on this extra phosphate onto ADP, allowing us to reform ATP. Very complex process. It's actually pretty beautiful if you get to watch an animation of it. But long story short, F0 subunit is allowing the protons through. F1 subunit is the one that is using that movement to slap on that extra phosphate. To summarize the reaction, every 4 protons that move down the gradient into the matrices is going to generate enough energy for us to recreate 1 ATP. 4 protons will create 1 ATP. So just as a reminder, NADH pumped 10 protons, and those 10 protons can therefore create 2.5 ATP per NADH. If you've seen that number before, that number is coming from the number of protons that NADH's electrons are pumping. FADH2, as a reminder, was only able to pump 6 protons. And so because of that, it is only going to be able to produce 1.5 ATP per FADH2. 6 protons pumped by FADH2, 4 per ATP, 6 divided by 4 is 1.5. Just some other information about oxidative phosphorylation. It is a form of what's called chemiosmotic coupling. This word chemiosmotic is basically just a term that's describing the favorable movement of ions down their concentration gradient, including protons. This favorable movement is then coupled to an unfavorable reaction, of course, ATP synthesis in the example that we're talking through right now, which is why it's specifically called chemiosmotic coupling. So ATP synthase, it runs oxidative phosphorylation, which is a type of chemiosmotic coupling. Those 3 things you should always be able to tie with each other. So there's 2 main ways to inhibit ATP synthase and oxidative phosphorylation. The first is to disrupt the proton gradient in any way. This is called uncoupling the proton gradient. There's a few ways we can do this. The most obvious one is by blocking the electron transport chain. If you block the electron transport chain, there's not going to be any more protons pumped, and therefore there's no proton gradient for ATP synthase to take advantage of. Additionally, any other process or reaction that decreases the hydrogen concentration of the intermembrane space, such as making the intermembrane space slightly more basic, or potentially using the proton, maybe there's some other cellular process that needs the protons for something. That is going to decrease the hydrogen concentration gradient, which is essentially removing this gradient that we need for oxidative phosphorylation. So uncoupling is the first way that we can inhibit oxidative phosphorylation. The other, of course, is just by inhibiting ATP synthase itself. You can block one of the subunits, you can do something to it random, it doesn't really matter. Basically, we have kind of the less obvious way of uncoupling the proton gradient from ATP synthase. That will inhibit, of course, but then, like everything else, you can just directly inhibit it if you want as well. If you shove a stick up, gonna be careful with my wording. If you slap something onto ATP synthase that's blocking it in some way, of course, that's going to inhibit it. We're going to come back to inhibition here in a few slides because we're specifically going to talk about regulation of the ETC and ATP synthase, when sometimes we can inhibit both, sometimes we can only inhibit one. We'll talk about that here in a short bit. Before we do that, I'm going to give a quick ATP production summary of aerobic respiration. Aerobic respiration is starting with glycolysis in the cytosol and ending with the Krebs cycle and electron transport chain in the mitochondrial matrix. Now that we understand how much ATP both of our electron carriers can produce, we can figure out how much ATP we're producing per glucose. So we're going to start with just the reminder that one NADH is going to pump 10 protons, which through oxidative phosphorylation will produce 2.5 ATP, so 2.5 ATP per NADH. One FADH2 will produce 1.5 ATP. That's why you'll notice, for example, right here in the Krebs cycle, 6 NADH is being turned into 15 ATP because 6 times 2.5 is 15. That's the first reminder. The second reminder is the general overview of the process, just to remind you of what we're producing in each of these steps. We're starting off with one glucose in the postprandial state when aerobic respiration is mainly promoted. This 6-carbon molecule is split up into 2 3-carbon pyruvates, which will allow us to produce 2 ATP directly and then 2 NADH that are going to need to get shuttled into the matrix, just as a reminder. The 2 pyruvate, which will also get shuttled into the matrix. Luckily, you don't need to know too much about that. These 2 pyruvate are going to run through the PDC, which will produce 2 2-carbon acetyl-CoAs. We've lost a carbon, which is why we have a carbon dioxide produced. And additionally, we're going to produce 2 NADH. Just as a reminder, everything we're looking at right now is per glucose. They might ask you per pyruvate. And so we're producing 1 NADH, 1 acetyl-CoA, and 1 CO2 per pyruvate, but we're talking about per glucose right now. Once we have this acetyl-CoA, we can run it through the Krebs cycle to produce 2 ATP, 6 NADH, and 2 FADH2. And that is going to give us the ending oxaloacetate and 2 carbon dioxides, but we don't really care about those anymore. We're pretty much done with the process. So starting over here with eukaryotic aerobic respiration. Just as a reminder, eukaryotes have membranes that we have to worry about. Our mitochondria has a membrane around it that we're going to have to shuttle pyruvate and the electrons from NADH over. And that's going to take a little bit of energy, like we've talked about with our shuttles. So in glycolysis, remember that this is per glucose. Glycolysis is producing 2 ATP and 2 NADH. I put a star right here because, as a reminder, the electrons from cytosolic NADH, they've got to cross that membrane through one of those 2 shuttles that can potentially lower our ATP yield. Those 2 NADH theoretically can give us a maximum of 5 ATP, but if the NADH both give their electrons to FADH2 through the G3P shuttle, we're only going to be producing 3 ATP. Again, that can be kind of confusing, but luckily for our purposes, the main thing to focus on is that we are producing somewhere between 5 to 7 ATP per glucose in glycolysis. Right? And just as a reminder, this is in the cytosol. Then we move on to the pyruvate dehydrogenase complex, where pyruvate being oxidized will help us produce 2 NADH. These 2 NADH are already in the matrix, so they can directly help us produce 5 ATP. So we're at a total of 10 to 12 ATP per glucose so far. So the PDC is directly helping us produce 5 ATP. Then we get to the Krebs cycle, where inside of the mitochondrial matrix, we'll be producing 2 GTP, which, like we've talked about earlier, are essentially the exact same thing as ATP. We'll also produce 6 NADH per glucose, which is going to yield 15 ATP at the electron transport chain, and 2 FADH2, which is going to yield 3 ATP at the ETC. So per glucose, we are producing 20 ATP in the Krebs cycle, 5 in the pyruvate dehydrogenase complex, and 5 to 7 in glycolysis, depending on which shuttle we decide to use. That's why our overall total ATP yield from aerobic respiration is 30 to 32 ATP per glucose if we add all 3 of those numbers up together. I've also added, just in a quick little gray box, that you do want to keep track of how many ATP each individual intermediate can make. So for example, acetyl-CoA is going to be making 1 GTP, 3 NADH, and 1 FADH2, which corresponds to 10 ATP if you start doing that math. And per pyruvate, we're only making 1 NADH in the PDC, which is going to go through and make 2.5 ATP. The MCAT can ask you either way. Just make sure you're understanding if it's asking for per glucose or per pyruvate, whatever they're asking. Just make sure that you're understanding which one they're looking for. Prokaryotes, however, they do not have any membraned organelles. They do still have organelles, like they have ribosomes and random things like that, but they don't have membraned organelles. And they also don't have mitochondria. So because of that, everything is just going to happen in the same place in prokaryotes. It's all going to happen in the cytosol. So because of that, prokaryotes do not have to worry about losing energy shipping something over a membrane. They're not going to have to lose any ATP yield from moving NADH into any mitochondrial structure because they don't have one and they don't have membrane-bound organelles. So because of this, you'll notice going through the exact same chart, everything is the exact same except for the cytosolic NADH. Because they do not have to move these electrons into some new chamber, they can produce the maximal 5 ATP per NADH in glycolysis. So because of that, we see that prokaryotes are able to produce 32 ATP per glucose. They essentially are slightly more efficient than eukaryotes. So now the thing that you might be asking is, okay, well, how does the electron transport chain work if they don't have an inner mitochondrial membrane like eukaryotes do? Well, just to kind of show you how this works, for prokaryotes, again, remember, like I said earlier in one of my earlier videos, that all of my slides are referring to eukaryotic metabolism unless I specified otherwise. This is where I'm specifying otherwise. Everything, for the most part, is going to be the exact same between prokaryotes and eukaryotes, except for these 3 bullet points. Like we just talked about on the last slide, prokaryotic ATP yield is going to be greater than eukaryotic due to not having to worry about a mitochondrial membrane. Again, prokaryotes do not have a mitochondria or any membranes around their organelles. So all of their metabolic enzymes and processes are just going to occur inside of the cytosol of the prokaryotic cell. So how exactly does this work with the electron transport chain? The ETC and ATP synthase, instead of being located on the inner mitochondrial membrane like in eukaryotes, they're actually just located on the plasma membrane of the bacterial cell. So they are essentially pumping protons out of the cytosol outside of the bacteria rather than pumping protons out of the mitochondrial matrix. So the cytosol of the prokaryotes is the region that we are pumping protons out of, making the cytosol the region that is going to be slightly more basic. The surrounding region around the prokaryotic cell will be slightly more acidic due to that growing hydrogen concentration. And then when they're ready to start making ATP via ATP synthase, they just let that hydrogen in through the plasma membrane on the cell. All right, keep holding on. I promise we're getting close. I know this is a lot, but like I always say, my goal is to tell you everything you need to know, and I hope that this is the only time you have to watch it. Hopefully you have my notes in front of you, so you already have everything written down. But now we're actually going to dive into a little bit of regulation of the electron transport chain and ATP synthase, because this is where the MCAT loves to sit and ask you questions. So we're going to start on the left with ways to inhibit the electron transport chain. Going to be a ton of text on this slide. My apologies for that, but like I said, it's all in the notes. Just sit and listen, make sure everything I'm talking about makes sense. So we're going to split this up into 2 different groups. We're going to start off by talking about what happens when you inhibit complexes 3 and 4. And there's a reason for this we'll get to in a minute. Because both NADH and FADH2 are sending electrons through complexes 3 and 4, if you inhibit complexes 3 or 4, that is going to shut down the entire electron transport chain. Kind of like we talked about earlier, where if we don't have oxygen present, then complex 4 can't reset itself, which means that cytochrome C gets stuck in the reduced form, which means that complex 3 gets stuck in the reduced form, so on and so forth. Basically, if you are inhibiting complex 3 or 4 in some way, that is going to halt the electron transport chain entirely. Neither complex 1 or 2 are going to be able to reset themselves. Neither of them are going to be able to accept electrons from FADH2 or NADH. For example, if you inhibit complex 3, it is not going to be able to pass on electrons to complex 4, and that's not going to allow complex 3 to reset itself to accept more electrons. This is going to cause that buildup of coenzyme Q in the reduced form, which of course, via negative feedback and buildup of products, is not going to allow complex 1 or 2 to work either. Basically explaining, inhibiting complex 3 or 4 is going to entirely halt the pumping of protons. Additionally, if you happen to inhibit both complex 1 and 2 at the exact same time, that is also going to entirely shut down the ETC. Complexes 1 and 2 are both of the entry points for electrons into the ETC, so it should make sense that if we inhibit both of them at the same time, even though it's unlikely, this is going to prevent any of the electron transport chain from working. No protons are going to be pumped. In this scenario where we have basically entirely shut down the electron transport chain, there's no protons that ATP synthase can take advantage of, and therefore the cell is mainly going to switch to anaerobic respiration, where we're only going to be running glycolysis, fermentation, and the Cori cycle, only producing 2 ATP per glucose. Conversely, if you inhibit either complex 1 or 2, but not both at the same time, this is going to decrease the strength of the hydrogen gradient, and therefore we'll see a little bit of a decrease in the total ATP yield, but it will not entirely shut down the electron transport chain. For example, let's just say that you inhibit complex 2 somehow. You inhibit complex 2, so FADH2 is no longer being oxidized. Complex 1 is still working perfectly fine. It's going to oxidize NADH, pump some protons, give those electrons onto coenzyme Q, forming ubiquinol, and coenzyme Q can then go on to complex 3. So while we might lose a little bit of our proton gradient production since we aren't taking advantage of FADH2, we're still able to pump some protons from NADH, and therefore we're still going to be able to produce some ATP. So basically, if you inhibit either one of complex 1 or 2, you're going to see a slight decrease in ATP yield, but not an entire disruption of the ETC. It's still going to work, just maybe not to the same extent. Again, ATP yield would decrease, but the cell can still do aerobic respiration and will continue to do so, just slightly less efficiently. And then, of course, since NADH and FADH2 are the start of the ETC, if we do any sort of process that's going to inhibit our NADH or FADH2 production, that is also in some way going to inhibit the ETC. If we entirely remove NADH and FADH2, the electron transport chain is going to be completely shut off. Basically, your brain should just be able to kind of think through what would happen if I shut down this complex, or if I stopped the cell from running this process. If it's stopping one of the electron carriers from being produced, we're going to see a slight decrease in ATP yield at the ETC. There's also a scenario like we talked about up here where you can potentially entirely inhibit ATP production at the electron transport chain through aerobic respiration. The ways that we can inhibit ATP synthase or oxidative phosphorylation. The first one should be a little bit obvious, because if you inhibit the electron transport chain in any way, this is also going to affect ATP synthase. If you decrease the proton gradient, that's going to decrease how much ATP we can produce. If you entirely shut off the electron transport chain and completely remove that proton gradient, there's no gradient for ATP synthase to do chemiosmotic coupling. So just connecting the ideas that we just talked about, if you inhibit complex 1 or 2, this is going to decrease the ATP production rate because there's less protons being pumped. Or if you entirely shut off the ETC by inhibiting complex 3 or 4, this is going to entirely shut down ATP production. There's not going to be any protons pumped. ATP synthase can't work at all. The cell is going to switch to anaerobic respiration. We already alluded to this, but uncoupling is another way that we can inhibit ATP synthase without doing anything to the electron transport chain. For example, maybe there's some medication that what it does is it raises the pH of the cytosol and intermembrane space. In doing so, that is going to decrease the hydrogen concentration, decreasing that gradient, and it is going to decrease ATP yield at ATP synthase. In other words, kind of like I'm trying to get at right now, it's possible that the ETC just keeps pumping protons, but there's something preventing ATP from being formed via oxidative phosphorylation. The electron transport chain's only goal is to pump protons whenever it can. So it's possible that the ETC is pumping protons just fine, but there's something going on with the proton gradient that is preventing ATP synthase from working properly. Conversely, you cannot inhibit the electron transport chain without it having some degree of effect on ATP synthesis. If you inhibit one of the complexes, that's going to do something to the proton gradient, which is going to do something to our ATP yield at ATP synthase. But you can inhibit ATP synthase without it affecting the electron transport chain. If you always connect the ETC to ATP synthase, you're just going to assume that if you inhibit one, the other one's inhibited. But that's not the case like we've demonstrated here. They are connected to each other through the proton gradient. So obviously, if the proton gradient is disrupted, then that's when we can maybe connect the ETC to the ATP synthase, but it's possible to inhibit ATP synthase without doing anything to the electron transport chain. All right, hopefully you're still alive, you're still with me, you haven't just completely fallen into oblivion. That is it for not only the electron transport chain and ATP synthase, but we're also done with aerobic respiration as well, which is a majority of the content you need to know for metabolism on the MCAT. So really appreciate you being patient with me. Hopefully this video has done a good job of explaining exactly what you need to know, nothing more, nothing less. And of course, as usual, if you need any other resources or videos, you can find them in the description in my bio.